Thank you very much for the chance to speak about this large project today. And we're going to talk broadly about BRD testing, which is kind of the back end now. We've had the prebiotic, probiotic discussion, vaccine prevention. And we're focused in this talk a lot about testing and how we see we might be able to integrate that into BRD management, knowing how complex that disease is and how important it is for the industry. Just a brief note of some of the disclosures for funding and sources here, and you'll see the research group that I lead, and I won't spend a lot of time here. If you've got more questions, please come and ask, but we're focused specifically on that Genome Canada, Genome Alberta, Saskatchewan Ag Development Fund project, Genomic ASSETS for Livestock. So briefly going to review antimicrobials and BRD management, which I realize is probably not new for most of you, but we're going to use that to funnel into this discussion about laboratory testing for the BRD pathogens and antimicrobial resistance. Talk about some of the limitations of testing, what we do and don't know about AMR with BRD, some testing opportunities linked to the project we've done, and a bit of a view to the future. So you'll see this slide again, talking about this large funded project, Genomic ASSETS for Livestock, and I will go through bits and pieces of that complex figure, but it's this idea about using sequencing-based technologies to test subsets of cattle from a pen at a feedlot to get a representative idea of what's going on in those pens and in those feedlots for bovine respiratory disease pathogens, antimicrobial resistance, and of course, as we talked about and heard in the last talk, knowing that those bugs are normal flora. So we see them and they're there. So we want to get an idea about what's going on in animals that get sick. And I'll talk more about these pieces as we go. And I do want to acknowledge Dr. Cheryl Waldner, who was the co-lead on this project. So it was Cheryl and I, together with a large team of researchers across the lab space, computing bioinformatics space, that conducted this large project. I'm not sure we really need to review the BRD slide. Thank you for the previous speaker, obviously, for doing that. But just knowing that we've got bacteria and viruses there, and that this is a disease complex, right? We've got a number of pathogens, a number of bacterial and viral, and this multifactorial disease. So we've got the animal factors, age, immune status, genetic susceptibility, the environmental factors, so transport stress, commingling, auction mart transport, those pieces. But we know that BRD, of course, is a large cause of morbidity and mortality in the industry and something that you deal with in that space. And we also know that it's the number one reason for injectable antimicrobial use. So my research is in AMR. We're concerned about antimicrobial resistance from a human health and an animal health standpoint. And from a One Health piece about understanding how these microbes move between these human, animal, environmental reservoirs, and largely you can maintain resistance reservoirs in that space. And we know that there's pressure on the industry to continue to demonstrate good antimicrobial stewardship. And that's why these research projects and the research that the beef industry has done in the AMR space over the last 20 years is so important. I've adapted these quotes, but it really it's where I'm going to start and where I want to take us back to. I think we can all acknowledge that BRD management is difficult. I think someone said this morning it's one of these challenges in the industry that we've been working on for a long time and have put a lot of money into research, but it continues to be challenging to manage, and antimicrobials are an important tool. I like the phrase, if you want to go fast, go alone. If you want to go far, go together. And I think that demonstrates the collaborative work in the beef industry with research, with veterinarians, surveillance, and government to continue to try to tackle this challenge. So we know that we use antimicrobial metaphylaxis on arrival, and that these protocols are based on BRD risk categorization. For example, our high-risk calves, recently weaned, auction source, fall place calves are probably going to get different metaphylaxis than older calves. When those calves get sick in the feedlot, they're pulled out of their pens based on clinical signs, fever, depression, labored breathing, not eating as well. And those treatment protocols, again, are based on risk category. And so we might use different antimicrobial drugs for first, second, and third treatments if there are recurrent cases. So these protocols are based on the data and consultation with veterinarians in the veterinary clinics. So they'll have these protocols that are doing this risk assessment on arrival. Those animals are given a metaphylactic drug if the protocol indicates that they should, and then we track them to see who is getting disease and who is not. And again, the treatment protocols are based on these risk categorizations, largely in consultation, of course, with the feedlot veterinarians. So let's transition to testing. We know when we're testing in this space that typically our standard go-to is bacterial culture. So in a lot of cases, we're taking a nasal swab, usually a deep nasopharyngeal swab under restraint in a chute, and we're going to send those swabs to a lab that's going to grow bacteria on some of the plates that we saw in the previous talk. So we identify which bacteria are present, and then when we're doing antimicrobial susceptibility testing, we look at the different drugs to see which antibiotics kill them and which ones don't, and we call this susceptibility testing or sensitivity testing. In some cases, we're doing PCR testing. So this is a molecular test that we're using that's identifying known targets for bacterial genetic material or viral genetic material. These are targeted tests, so you can only find the bugs for which you have known targets. And in a lot of cases, in both of these spaces, we're talking about targeted testing. Can we identify certain organisms or genetic complements that tell us that the organisms are present? So you can't find the bug if you don't have a specific test for it or if you aren't looking for it. And so when we look under the hood, the list of common and available tests is relatively short. So we equate traditional testing as kind of like fishing with one hook at a time, where we're looking for specific things. We know that there are big limitations to testing and that routine testing is widely not used for BRD management for these reasons. Typical turnaround time when we take a sample to get a result back to you as a producer or to a veterinarian is 5 to 7 days. In some cases, we've got shipping distance issues from the feedlot to the vet clinic to the lab. We have lab processing time, and just reinforcing we don't have chute-side tests that work for BRD in terms of laboratory tests. I know there's other technology out there that is less in my space, looking at heat thermography, using stethoscopes on chests, and so those are things to consider. I'm talking about these laboratory bug tests. When we talk to people who aren't well versed in feedlot production and they ask us about testing for individual animals to inform treatment, we say we would need an answer within seconds in the chute, right? You cannot wait longer than that. So 5 to 7 days, obviously, for routine testing is completely out of the realm of possibility. It's useful from a surveillance perspective when we can track these things over time. So we have remote feedlots, remote from clinics, diagnostic labs. Shipping conditions can also affect our sample quality. If we have those nasal swabs that are taken at 30 degrees and they're left on the dash of the truck, or if they get stuck over a long weekend shipping at -40, that is going to impact our ability to culture and our ability to detect genetic material. So these delays reduce usefulness for acute treatment, obviously. So I think it goes without saying that it's not practical to test every sick calf before metaphylaxis or treatment. There's a cost involved, there's time, and ultimately we know that there's animal health and welfare benefits if we delay treatment. So one of the questions we're asking with these projects are, can we use group level testing to have group level data to inform future treatment decisions? If we look at the current data for BRD pathogens based on the successful Canadian Feedlot AMU-AMR surveillance program that's conducted with funding from the organizations we've seen today, in collaboration with the Canadian Integrated Program for AMR Surveillance, we know that the levels of resistance are not that high in what we're seeing in the pathogens that are identified. So veterinarians, their technologists are collecting respiratory samples from commercial feedlots in Western Canada. They're tested for phenotypic resistance in BRD pathogens at arrival, and then they're sampled again later in the feeding period. Variable, sometimes 60 to 75 days, usually at a time when those cattle are coming through the chute again. So from this, we have regional, temporal trends in BRD pathogen and AMR prevalence data, and they're also tracking antimicrobial use. We can look at the Mannheimia hemolytica data taken from the CFASP website in 2024. Those yellow bars are the arrival samples, and the blue bars are the rehandling samples. And it demonstrates a fairly common phenomena that the resistance on arrival is generally very low to negligible for most of the drugs tested. And we can see on that figure that the macrolides and oxytetracycline levels of resistance tend to go up in rehandling because of the selective pressure of the metaphylaxis drugs that are used. Just another look at this, confirming that resistance to common BRD drugs is generally low, and it increases short-term after metaphylaxis. These graphs show the number of drugs to which those different BRD pathogens are susceptible or resistant to. So you've got arrival and rehandling for both of those. So Mannheimia, Pastorella, Histophilus, arrival and rehandling, and then calf versus yearling. And so we can see that the numbers that are orange, gray, and yellow, which are resistant to 1, 2, or 3 or more antimicrobials, tend to be higher at rehandling than they are at arrival. But again, the prevalences are still not very high. So this is part of one of the sections of one of the studies that we did. So the first 2 years of fall trials were conducted at the Livestock and Forage Center of Excellence at the University of Saskatchewan. So the research feedlot there, and so there we collected a number of respiratory samples, deep nasopharyngeal swabs from these calves. So there were 400 calves in 2020, 400 calves in 2021, placed in groups of 100 in pens, and samples were taken at day 0, day 13, and day 36 after arrival. This is the phenotypic susceptibility testing, so that traditional culture, and it's just meant to, again, to demonstrate this change in resistance over time. So if you look at those graphs, we've got the fine dots represent day 1 samples, the dashes day 13 samples, and the solid lines day 36 samples. We've got 4 different macrolides here, so Draxin and Zactran on the left, Soprevo and then Micotil on the right. And what we see, if we look at these concentrations on the bottom, so that the concentration level indicates a level of susceptibility or resistance. So if you look at these lines, that concentration, if detected, says that that's a resistant. So the higher the number, the more resistant the organism. And we can see for these Mannheimia isolates that the day 13, day 36 isolates have a much higher distribution of MIC compared to the ones in blue, where we have a much higher distribution for the lower MIC. So again, demonstrating this phenomenon of increased levels of resistance after metaphylaxis. And again, just some more data showing this across the 3 organisms from the federal surveillance program, seeing that these blue bars in Pastorella and Mannheimia go up rehandling. It's a little bit less distinct in the Histophilus somni isolates. What's interesting about AMR and BRD, and I'm curious to hear what Steve and others think in the audience, is that we're still not clear if AMR leads to worse BRD treatment outcomes. When you look at the literature on this, and Cheryl and I did a deep dive into it, there's actually very few studies that assess this link. What are the treatment outcomes in these animals that actually have resistant pathogens? And the few that do look at it either don't show that there's a link or the results are inconclusive. But I always use the phrase, absence of evidence is not evidence of absence. And I would suggest that this is a knowledge gap that we need to continue to look at. The perception that I've heard from talking to people is that these BRD drugs may not be working as well as they used to be. And I'm not sure if that's in terms of metaphylaxis or treatment. I can't comment. I'd be curious to know what people think. But as I said, I think we've got a lack of literature that really looks at treatment outcomes. And so this leads us into the questions about, can diagnostic data be generated prior to treatment decisions that can be helpful at a pen or a feedlot level? What tests can we use? Who do we need to sample to inform BRD treatment decisions? And when is an informative sampling time relative to disease onset? And this leads us into the testing opportunities that we've been working on evaluating through Genomic ASSETS. So as I mentioned, the idea is, can we take a pen of feedlot cattle or a lot coming in on arrival, take a representative sample from a number of those calves? In this case, what we really sought to achieve with this project was to use long-read metagenomic sequencing. So the idea that we're taking a nasal swab and we're sequencing everything in there. And without getting too far in the weeds, the technology that we leaned towards was called nanopore. So that's what that little funny little green and black thing is, and it's the sequencing piece. But the part I want you to take away is when traditional whole genome sequencing, usually the DNA is all broken up into tiny fragments. And so we send that away, we sequence a pure culture of an isolate, and they send us back sequences of all these little chunks. And then we have to do this computing step where they call it alignment, where we assemble those genomes against a reference standard to say, based on how we put all those pieces together, we're pretty confident that this is this. With long-read sequencing, we get much longer pieces of DNA, and I'll talk about why that's important in a minute. But you can imagine there's an intense computing requirement in the background, and that was one of the big development pieces of this project, was to develop the pipeline based on the protocols we designed to be able to identify the bugs and the resistance genes, but also to process the genomic data from thousands of samples. We've collected probably in the order of 3,000 to 4,000 samples across this project. And at one point, we were taking a large, substantial portion of the entire U of S server capacity with something like 2 terabytes of data. So can we use that, especially at different time points, to understand what resistance started at, what it changes to, and then in some of the analyses done in this, looking at associations with who gets sick, and ultimately tie these data back to veterinarians to say, do we need to look at our BRD treatment protocols based on levels of resistance? So being really clear, we don't have anything quick enough for metaphylaxis right now. We might be able to use this year's data for something next year, but we're really focused on these treatment protocols. So as I mentioned, metagenomic sequencing is cool in that it reads all the DNA in a sample. So if we take a nasal swab from a calf, we're going to sequence everything that's in there. It's not specific. We're not doing culture specific to an organism. So it can identify things that we know, as well as things that were previously unreported. And it's a single sequencing run that can tell you about everything in that sample. Now we have to do some fancy things to worry about the cow DNA that's in there, and the protocol manages that. The other thing that we found in this is we needed to do a non-specific enrichment step. So we took those swabs, we put them in culture media for 10 to 14 hours and just let things grow. So compared to the previous talk where we're looking at the microbiome, we don't care about the microbiome. Not that it's not important, but that's not what we're trying to learn about. We're trying to figure out whether or not we can figure out whether these bugs are present or absent and whether or not they have resistance genes. And what's cool about it is we have all these data, so we can run the computing today and find what we know, and then when we know new things and new genes 2 years from now, we could go back and run it again to see if we can find it. So think of it like a net. We're netting for everything rather than using a fishing hook and a fishing rod that looks for one specific thing. And I'm pretty sure this slide pretty much just confirms the same thing. Conventional culture in PCR is targeted for bugs and known genes. Sequencing can find everything that's there. Now, no test is perfect. We know that. In the validation work that we did through these large field studies, research feedlot, and the commercial feedlot study, we found that we had tests that were comparable to traditional culture for identifying BRD bacteria. We had to do that enrichment step to find the resistance genes. The metagenomic sequencing identifies samples with resistance genes with similar performance to these culture-based tests, but it can identify many more types of bacteria and resistance genes with a single test or a sequencing run. So there's efficiency there. We were finding pink eye bacteria, we were finding foodborne bacteria, indicators. We can do a lot of different deep dives into that. We think that there's potential for quicker turnaround times with this once operationalized, but we're still limited by those bottlenecks of shipping time and things like that. That's something that we don't have an easy solution for. And we experienced that firsthand working with the vet clinics and trying to coordinate sampling earlier in the week so we didn't have samples sitting in shipping trucks over the weekend. So we're really grateful to have had the opportunity to work with feedlot veterinarians like Steve and their clinics and their staff to do the field study as part of this project in 2022. Because of the positive linkages with CPARS and CanFASP, is we were able to piggyback on their already existing sampling and surveillance. So they collected extra swabs for us, and then we had access to their susceptibility testing data so that we could compare our new metagenomic detection to the susceptibility data that CPARS had. And that's one of the positives of the strong ongoing collaboration between surveillance veterinarians and all of you as producers. So they recruited 19 separate feedlots. They picked one pen from each feedlot. They collected 20 samples per pen on arrival and at day 14. So there were 13 pens of fall-placed calves and then 6 pens of yearlings. And so again, we had PDS that was doing all of the testing. They did the library preparation, and then the sequencing was done at an affiliated, that's the Opal lab at U of S that did the nanopore sequencing for us. Don't worry a ton about all the details on here. I will walk through what's important, but it's this idea about pen level testing. So collecting and testing swabs from a subset of 20 calves per pen at 1 and 13 days on feed, determining the prevalence of calves with each of those bacterial pathogens as well as resistance. And then we did a lot of thinking about what would trigger a change. And I don't know that we know what triggers a change in terms of resistance prevalence, but just knowing that if we feed that data back, maybe that prompts a change in a treatment protocol if we exceed that threshold. And these data flow through the veterinarian and back to you as a producer to make some of these decisions. So what did we learn from long-read metagenomic sequencing? We know this already, right? There's many different bacteria in a healthy calf's nose and in most healthy calves, and that respiratory bacteria that can cause BRD are present before disease. They're bugs that we typically culture, but we've got more data now. So we can tell you different serotypes of Mannheimia hemolytica that are there based on the metagenomic sequencing. It's not something we can do at all with culture easily. You can do some PCR testing, but again, this is a one step here that we can do. So with the sequencing and the databases, we can tell you the serotypes that are there. We can find some of these hard to culture bugs like Mycoplasma bovis, Mycoplasma dispar, and other things that we're realizing might have importance that haven't been part of our traditional core. What's really cool about these long read sequencing data in the bioinformatics is that when you read a lot of metagenomic studies, they say we did metagenomics on this sample. These bugs were here, these genes were here, we have no idea which bugs had which genes. With the long-read sequencing, with the bioinformatics, we're able to link resistance genes to specific bacteria. So we can tell you that Mannheimia had this resistance and Pastorella had this. We can identify serotypes, and we can also identify mobile genetic elements within there too, like ICE elements that are responsible for spreading these genes around. So that's a strength, I think, of the enrichment, of the long-read sequencing, and then of the bioinformatic and computing pipeline that we have as well. The other cool thing that we've done, which hasn't been published yet, but we had the opportunity to do a different type of sequencing from a different company that replicated and confirmed that they were getting the same results from PacBio as we were getting with the nanopore sequencing, which is also cool. So we have an unlimited ARG listing. We've got these databases that are updated all the time with new resistance genes, and we can look for those things with the sequence data and the bioinformatics. So it reveals many resistance genes in a single run. As I said, it's linked to the bacteria of interest, which is a novel finding for what we're doing with metagenomics. We can see multiple resistance patterns co-occurring sometimes within the same organism, and we can find these mobile genetic elements. As I heard Cheryl say in a talk about this last week, we like to get greedy when we do these things, and we also had the ability to modify that protocol to go and look for respiratory viruses as well. So we had the veterinarians and their technologists collect a third swab, and we could detect many viruses that currently have no available PCR tests, some of which are linked to disease, and we validated some of this with PCR detection. So we found common viruses, IBR, BVD, BRSV, PI3, and a whole number of other ones there as well, coronavirus, some influenza D, and there's a paper published on that, and if you want, I can send you the links for it. But what's really interesting about the viral piece is we might be able to use this as a test to look at vaccine prevalence, vaccine efficacy in terms of detection of viruses using this sequencing technology. So to be clear, we probably can't do the bacterial and viral on one single swab. We might need two, but we have the protocols to be able to do those things. So going back to what we know and what we don't know, and there's a lot of epi-analysis that's been done in the background here, but we've been able to show through the different time points using sequencing and phenotypic data that, in the sequencing side, metagenomic detection of Mannheimia hemolytica and Pastorella multocida at day 13 had increased risk of BRD at days 14 to 45. Detection of macrolide or tetracycline AMR genes at that day 13 time period had increased risk of macrolide or tetracycline resistant BRD bacteria in sick calves at days 14 to 45. And further analysis to show that yes, individual detection is associated with individual risk, but that pen level detection can also help us infer individual risk as well, which kind of gets to that pen level sampling. I think what we found was that because resistance was so low on day 0 that it wasn't helpful for us. It was that kind of 2-week time point of sampling that was more informative, recognizing that that's not necessarily a great time to have to run cattle through the chute. So we know that AMR is dynamic. It changes with antibiotic administration and time. But we've learned in this that sampling calves before clinical disease can anticipate resistance risks. Sampling at 2 weeks on feed was more informative than arrival sampling alone, and that pen level sampling results are linked to individual BRD risk. And this idea that long read sequencing can do this detection of things because of these long reads. So we're starting to get some more clarity about who we need to sample, that a subset of calves in a pen is helpful. Early in the feeding period worked in this sense, 2 weeks on feed, but I think there's still work that needs to be done to find that sweet spot. But again, the what of it, that culture or metagenomic sequencing seem to be, the sequencing I think is going to give us an option there and something that can give us insight into BRD treatment risk. So we know that bacteria that cause disease can be found in Morse normal calves. When recently weaned fall-placed calves arrive, there's very little AMR, but it increases in that post-metaphylactic window. The other thing we saw because of the sequencing we were doing is that some of these strains are contagious. And one of the feedlot studies, in the research feedlot, they had a contagious resistant strain that we watched spread through multiple calves. And metagenomics gives us this deeper look at bacteria, AMR, and viruses than any other single test. Without a doubt, more work needs to be done on commercial validation, the feasibility of pen-level testing, the effects of sampling and targeted handling on welfare, stress, and transmission risk, especially thinking about day 14 when they're not necessarily in the chute, but really figuring out that optimal timing sweet spot. And then, again, carrying this forward to investigate health, welfare, and economic benefits from outcomes using testing, linking pen-level resistance data to treatment success rates, and having these conversations about what an actionable pen-level threshold might be. How much resistance do you need in a pen before you might change a treatment protocol? So this was some of the work that I led. There's some detailed pieces here. Don't worry a ton about it. I'll give you the highlights. But we were trying to map out information flow in the feedlot for bovine respiratory disease, production processes, information and information processing. And it sounds a bit heady and weird, but we use this value stream mapping, which is a manufacturing process thing where you look at manufacturing efficiency, but others have used this information quality dimension for biological processes. So we worked with people knowledgeable in the industry to map out BRD information flow to really realize we're not using testing in most commercial settings just because of all the limitations I've mentioned. But knowing what we know now about culture and metagenomics, these little blue squiggles are areas within that space where we felt that maybe testing could give us information here that we didn't have. But again, it's about making pen and feedlot level decisions, not individual animal decisions. My PhD student, Dr. Otto, and we're very grateful to the collaboration with the feedlot veterinarians, where she did a qualitative interview study to talk about diagnostic testing, to understand how it's being used and barriers for implementation of use, and a couple of things confirming what we know, right? The barriers about timing, the shipping delays, those were issues. But ultimately, anything that gets put forward here needs to have demonstrable benefit for you as producers in terms of animal health and welfare and ultimately economics. So where do I think this is going? My pie in the sky? I think what's great about metagenomics is we're not limited by the need for pre-identified targets. We can detect hard-to-culture organisms, and we can reveal other unexpected resistance patterns, and just the ability to continually interrogate data. I think there's hopefully possibility for integration into surveillance, to move from culture-based sequencing, culture-based to sequencing-based surveillance. There's been some interesting discussions with collaborators like Tim and Rahat at AAFC about pooled sample sequencing, and some, getting trying to see how we can make it even more efficient rather than trying to do all these individual swabs. I don't know where artificial intelligence is taking us in this space. I'm by no means an AI expert, so this is me kind of guessing a little bit here, but having heard a few other talks, I think that using computer learning tools to start to predict resistance based on sequence patterns and gene associations. So as our sequencing continues to get better, can we use computers to say, maybe we're not seeing resistance, but what do these gene associations mean, or gene regulation? And the question about linkage to animal genetics, it's not just the bugs, we've got the host part of it as well, right? The increased speed of computing and AI tools, using AI to check and generate code for sequence processing. I think these are all uncharted frontiers, and again, I'm not the expert to talk about it, but it would be naive to ignore that it's coming in some shape or form. So this is where I take us back to our quote, BRD management is hard. I think that testing is going to hopefully, should have had an arrow to helpful there. I don't know why I just deleted that, but I think the danger with high-powered computing is this: if you want to go fast, go alone. Let the computer tell us the answer. I think these things can be helpful for us, and if we link them up, that we continue to work together and work towards solutions in this space with this idea that pen-level testing is something that might be useful for us moving forward. So I really appreciate, there's my arrow. It just popped in when I hit the button. There. Thank you very much for your attention. I'd be happy to take any questions.
All right, is there any questions on this basic, simple research you're doing?
One of the things that's always confused the heck out of me is the difference between the genetic and the phenotypic pieces. So it was really actually kind of neat to see some of those being linked together, because a lot of times you can have the gene and maybe not have that actual phenotypic resistance happening, which may explain some of the treatment outcomes, right?
And so when you look at the papers, and I can send you links for anything you want, it's the genetic detection was still linked to actual BRD, clinical BRD in the research feedlot study, as well as resistance. We know that the link between genetic detection and phenotypic resistance is not always great, and there needs to be more work there, but the epi analysis that we've done actually shows that there's linkages there. Now, we couldn't do that in the 2022 study because those weren't animals that we were privy to all of that information for, but I think those are some of the opportunities to look at that moving forward.
Brenda's already asked lots of questions. You can go second.
Yeah, similar to our question, metagenomic, we know the DNA can be present, but the organism is not. How do you bring that into? The DNA can be present in that environment, but not necessarily the organism. But
that's the point. We are linking it to the organism because of the long-read sequencing. So we know with confidence that those genes are on those pathogens because of the long reads and the bioinformatic tools that we're using. That's the big difference with the sequencing protocol that we've used. To be clear, we had to do enrichment to do that. The first time we tried it without enrichment, we could find the bugs, but we couldn't find the genes very reliably.
Hey, Simon, back in 2019 when we were working on I can't say it. That's why I don't work in it anymore. Antimicrobial resistance in the feedlot and doing the deep nasal swabs, which aren't a hell of a lot of fun. And Lynn looking at the water bowl swabs. And I know Murray Jelinski had looked into that more. And if we talk about having to do a scan at day 14 and maybe doing it every week or whatever, where are you at with the water bowl swabbing?
Yeah, I think it's something we need to look at, right? Can we do environmental? I mean, it's the pooled sample as well, right? Do we need individual calves, or can we look at other environmental pieces for sure? We know these things can be contagious when they're there, not always, but yeah, those are all things that need to be looked at. We haven't done it here, but we should be. And again, is that enough to help us inform those pen level, feedlot level protocols, right? Do we necessarily need individual pieces, or individual animal samples?
Okay, Dr. Joyce has a lot. Are nasal isolates from healthy calves the same as the isolates that cause fatal disease? If not, then how do you interpret AMR findings from nasal isolates of healthy calves?
I'm trying to think about that. I mean, so when these samples were taken, we weren't distinguishing who was healthy and who was sick at the time. So in the 2022 study, we just got samples. We didn't know who was who, but they were coming out of the pens. So whether they were sick or not, we don't know. We were able to track that in the research feedlot study at the U of S. But day 0, they're all in theory going to be healthy, or if they weren't identified as sick. And then we tracked who got sick. So there's analyses in those papers that looks at the sick calves relative to healthy calves. But can we look at it and say they were always different bugs? I'm not sure that I've got the answer to that.
Current veto research with H. somni shows that the strains are genetically different, healthy versus fatal disease isolates. Does the presence of resistance genes mean the animal won't respond to the antimicrobial? Are genes turned on?
Don't know the answer to that, especially in H. somni. I know that we've done they were using the RPA test in this as well, and I haven't talked much about that today, trying to figure out if they could identify virulent strains. We had some trouble with that for sure. I think the serotype piece on the Mannheimia side is helpful. Just because they've got serotype 1 and 6 doesn't mean it's necessarily going to always be sick, but more likely to be sick. But there were definitely some associations between serotype specific detection and BRD and AMR in some cases, I think.
Okay, last one. How will this actually change how we treat cattle? We know if we get AMR if we know we get AMR with use of that drug for a short period anyway, we have limited classes of AM to use.
I think that's the big question, right? Like, we talked about actionable thresholds, right? About would we get to a point where resistance is high enough that we would change something knowing that we don't have a lot of treatment options? And that's a tricky question, right? Cheryl has got some good modeling papers for anybody who really wants to get in the weeds, where she's developed a simulation model about resistance movement through feedlots. The reality is right now, because resistance levels are so low, based on the simulation models, it's not having an impact on outcomes. But when you do sensitivity analysis to make resistance really high on arrival in particular, you can start to see treatment failures in a mathematical model. But we still haven't figured out what that looks like in real life yet. You know, as I said, please chime in here if there's a perception the drugs aren't working as well. That's what we're hearing, but we're not seeing it in the surveillance, recognizing that the surveillance is not everybody. It's a group of herds and feedlots.
So to your point there, and this is just a very boots on the ground perspective, the drugs are not working, and I have this conversation multiple times with my dad and with our vets. Have the post-treatment intervals ever been revisited? Because when Draxxin, for example, first came out, it was 14 days. Now they say it's 7, but being boots on the ground, it is not 7 days.
Yeah.
So all I'm getting at is when we see those animals are sick, we're pulling them and we're treating them. But I think if those windows were revisited and changed, I think our use of antimicrobials would go down, because when we're waiting those days, the damage to the lung is already done. So then we're behind on that sickness. That animal becomes a chronic and usually ends up on our dead pile. Right.
Yeah, and I mean, and that would love to have the sequencing data on those to see that, right? I mean, those are areas that we definitely need to look at to see what's changing for sure. I don't have a good answer for you, I'm sorry, but I really appreciate the context, because you hear that, but hearing it direct from boots on the ground is really helpful.
You know, just to add to that, animals not responding, I sometimes wonder if it's more isolated than at a pen level. Say, for example, a calf that received a drug at weaning time or at branding time or during his calfhood life before he ever came to the feedlot. Maybe specifically he's not responding, but at a pen level you may not pick that up. And on a research scale, there's just not enough of those specific animals showing up.
Yeah, no, that's
to see that.
That's helpful to hear as well, right? I mean, I think we've targeted the pen level piece, knowing that trying to make individual decisions based on treatment just isn't feasible until we have a test that's quicker, for sure. But you're right, like those more specific situations, you're still probably going to miss at a pen level.
Hi, really nice presentation, thank you.
Thanks.
I wondered if you have ever ran into any issues with computational power. I've done some meta-analysis and that was my issue all the time.
Yeah.
So especially if you're looking into rolling that out to more like a surveillance tool, I just wondered if you have any thoughts on that and how you would deal with that.
We definitely wrestled with that a lot in the project, and we had the benefit of working with some really good computer brains. So Paul Stothard at the U of A, and one of his postdocs was the one who developed the pipeline, and then we took that over to U of S to their high performance computing system. But yeah, we bumped into that. We bumped into computing power issues. Not that it wasn't there, but it's who's next in the queue, right? And I mean, as researchers, we have access to Compute Canada. I can't remember if that's still what Digital Research Alliance cluster, but computing power is one. And then I mentioned the storage issue, right? Like, we're chewing up huge amounts, and then you've got to figure out where you back it up. So interestingly, we are working on a grant right now in Alberta with Genome Alberta to look at coordination of genomic surveillance across human-animal environment. And one of the things we're proposing to do is just map those resources. It's one thing to know what tests and what data we have, but what computing resources do we have? What would we need to make sure that these things can go? Because it's always going to be a bottleneck. And now we're competing with AI for that bottleneck.